Okay, we're going to continue on with the respiratory system. And now that we've gone through all of the anatomy, we're going to talk about the mechanics of breathing. So, when we talk about breathing, we're going to look at two different competing pressures. We've got atmospheric pressure, which is obviously the pressure in the atmosphere, and intrapulmonary pressure, which is the pressure down in the alveoli. So, atmospheric pressure at sea level is 760 millimeters of mercury, or what's known as 1 atmosphere. So we're going to work off of that. Intrapulmonary pressure is something that we can change. We're not in control of atmospheric pressure, but we can change the amount of pressure that's in the alveoli and either make it above or below atmospheric pressure. The lungs have a significant number of collapsing forces in them. So if we think about, first of all, it's filled with elastic tissue. Picture a rubber band. So a rubber band doesn't want to stay in its stretched out state. Unless you force it to stay in the stretched out state, it's going to recoil. So the lungs are consistently wanting to recoil in on themselves. Then we've got the collapsing forces within the alveoli themselves. So if you picture the alveoli as kind of like a little balloon, okay? Running along the inside of that balloon is a very thin film of water. Now, water is cohesive, which means water likes water, okay? And so what they want to do is the walls of the alveoli want to collapse in on themselves because the water wants to be next to the water, and so it's attracting it. So these two forces, the fact that the lungs themselves are elastic and want to recoil, and the fact that we've got the cohesiveness of water that we have to overcome, means that we are constantly working against the collapsing forces. In order to work against them, we have to utilize the chest wall and the diaphragm significantly. And this is where we get into the pleura and the role that they play. So remember that the parietal pleura is running all along the rib cage and the diaphragm. So picture yourself breathing and you can feel your chest wall expand. So we take a big deep breath and everything's going to expand. When you pull on the chest wall, when you activate the muscles that are pulling on the chest wall and pushing the diaphragm down, what you're doing is you're pulling on the parietal pleura. Now, like we talked about before, the parietal pleura is in contact with the visceral pleura because there is a significant amount of surface tension between the two because of that layer of fluid between them. So when you pull on the parietal pleura, the visceral pleura comes with it. And then the visceral pleura is attached to the lungs. And so the fact that your chest wall does not cave in means that you are essentially always pulling out just a little bit on that lung tissue via the pleura. So picture you're doing dishes. Everybody's done dishes, hopefully. If you haven't, you should try it. It's a good character building experience. And you've had two plates stuck together in the soapy water. They're stuck together because of the surface tension in the water between them. That's what your pleura are like. So when in that moment, you can't move one plate without moving the other. They are stuck together. That's exactly what we want with the pleura. We want them stuck together by the surface tension between them. Now, go back to your plates. We don't always want our plates to be stuck together. We'd like to clean them each individually, and so we have to get them apart. And what we usually do to get them apart is kind of try to get something in between them to introduce some air. And as soon as air moves in between them, the surface tension is broken and the two plates fall apart. If this happens to your pleura, it's called a pneumothorax. It's the introduction of air, blood, pus, anything that interferes with the surface tension between the two layers of the pleura, and it causes that exact thing to happen. It causes the two layers to separate. Now, when you move your chest wall to breathe, the parietal pleura is moving with it. but the visceral pleura is no longer attached, and all the collapsing forces in the lungs are allowing the lung to collapse inward. Pneumothorax is the cause of collapsed lungs. So that's a very good illustration by what we mean, that if you're not consistently opposing the collapsing forces, the lungs will collapse on themselves. And then it requires a significant amount of energy to get them inflated again. If you have a pneumothorax, we have to put in a chest tube and we have to drain out whatever is interfering with that surface tension so that the two layers of the pleura can come back together. So yes, the pleura are going to reduce friction, but they also have a really important physiological role. Literally, you cannot breathe if they are not functioning properly. All right, so ventilation is the movement of air in and out of the lungs. And ventilation follows what's called Boyle's law, which states that volume changes lead to pressure changes, which lead to the flowing of gases to equalize pressure. So when you think of gases, you want to think of the same sort of concept as a concentration gradient, except it's now called a pressure gradient. So again, everything wants to come to its lowest possible energy state, and so if you have a high pressure in one location and a low pressure in another, and the gases can flow, then it will flow from high to low until we reach equilibrium. So in terms of our lungs, we can't force gas to go in one direction or the other, and we can't just change pressure without doing something. The thing that we can control in terms of Boyle's law is volume. We can change the volume of our lungs, and changing the volume leads to the pressure changes. We've seen this exact thing in the heart, where we talked about contracting and relaxing and how it changes pressure. So in terms of what we are responsible for in breathing and ventilation, our job is to contract or relax the muscles that change the volume. That then leads to changes in intrapulmonary pressure, which is going to take it above or below atmospheric pressure, which leads to gases passively flowing down their pressure gradient. So during inspiration or inhalation, we are going to contract muscles. Inspiration is always active. We have to contract the diaphragm, and it pulls down. It makes the lungs taller. It increases the height of the lungs. And we contract the external intercostals, which pull our rib cage out, and that's going to increase the width. So if you think of your lungs as kind of rectangles, when you are inspiring, you are going to increase both the height and the width, which is going to increase the volume. That is going to decrease intrapulmonary pressure to somewhere in the neighborhood of 1 to 2 millimeters of mercury below atmospheric pressure. So we're talking about somewhere in the neighborhood of like 758, 759. That's a very small gradient, but it's a gradient. Air will passively flow into our lungs. We don't want a huge gradient here because if air flows in too fast, it can damage the tissues. So we just need a small gradient. So all of that is active. I've got to contract the diaphragm. I've got to contract the external intercostals. Expiration is generally passive. You've used all your energy to pull the rubber band out. If you want the rubber band to go back, you just let go. And that's what we're doing during expiration. You are relying on that elasticity in the lungs to recoil, which is going to decrease the volume, which is going to increase the intrapulmonary pressure to a little bit above atmospheric pressure, and air is going to flow back out. So we don't move air. Our job is to change the volume. That leads to the pressure changes, which leads to gases flowing. Now, sometimes we'd undergo forced ventilation, so if we're breathing very deeply or quickly. So in terms of a deep or forced inspiration, we're going to take an active process and make it even more active. We're going to use a lot of the muscles in our neck, so the scalene, sternocleidomastoid, erector spinae, pec minor, it's going to pull up on the rib cage, and all of these are going to make it so that we can make the volume even bigger. That's going to drop the pressure even farther, which is going to allow us to have a larger gradient, and more air is going to flow in order to reach equilibrium. We can also do forced expiration. So here we're going to take a normally passive process and turn it active. And to do this, what we're doing is we're going to contract all of our abdominals and push the organs up against the diaphragm, which is going to have the effect of shortening the lungs. We also contract the internal intercostals, which pulls the rib cage in, and that's going to decrease the width. So we can absolutely change the amount of air that's moving in and out, but again, what do we do to change it? We change volume. That's what we're in charge of. Here we can see some of the muscles that are involved, so you can kind of look through these and see the different ways that this is going to work in terms of either increasing or decreasing the volume. So we've got some things that are going to influence ventilation. First of all is just the amount of resistance that air is going to encounter as it flows through the tree. So this is the same thing as resistance to blood flow that we talked about. You have the most medium-sized bronchi, so it's going to encounter the most resistance there. Now, we can't significantly change resistance aside from bronchodilating, but generally that is only really going to happen during a sympathetic nervous response. The other things that are going to play into this are the surface tension in the alveoli. So again, that very thin layer of fluid is mainly water, and that cohesive nature of water is going to make those walls want to collapse in on themselves. So in order to decrease that, this is where our surfactant comes into play. So we said that surfactant was something that was secreted by our type 2 cells. Surfactant interacts with water and decreases the amount of, or the sort of drive for water to interact with each other. And so it's overall, it's going to decrease the cohesiveness of water, which is going to help allow us to make sure that air can get down into the alveoli so that it remains inflated. Pulmonary surfactant is really important in the function of the lungs, and that can be illustrated very clearly in what's called infant respiratory distress syndrome. If you have premature babies somewhere around 35 weeks or less, a lot of times they are born with significant respiratory issues, primarily because the type 2 cells do not begin to synthesize and secrete surfactant until much later, until we're in the 36, 37, 38, 39, 40 week mark, because the lungs aren't inflated. So there's no reason for the baby to produce all of this if it's not supposed to be born for another month. So this is something that we see in premature infants, and unfortunately a lot of them end up on a ventilator right out of the gate, and that is correlated with lifelong respiratory problems. So if we can avoid this, we absolutely try to. We will give the mother steroids to try to speed up development, put them on bed rest. We will do anything we can to try to keep the baby in there as long as possible until we get past the point that the surfactant is going to show up, because without it, it is almost impossible for the baby to inflate its lungs. Last thing that's going to influence ventilation is going to be the compliance of the lungs. So in other words, do they do what you're asking them to do? So we have to have distendability in the lung tissue. We have to have the surface tension within the alveoli decreased enough that we're able to actually inflate. Here is where different types of diseases come into play that we can see issues. So something like emphysema dramatically decreases the elasticity of the lungs, which is going to decrease distendability. And so even when you are asking the lungs to inflate or recoil, it can't. because you don't have the elastic tissue anymore. And then the thoracic wall distendability will also play into this because obviously in order to breathe, I need the thoracic wall to pull out on the lungs. So if I have something, so here's a temporary example, when you have broken ribs, it hurts very much to contract, and so you tend to breathe very shallow. If we're talking about something that is more permanent and we talk about something like ALS, where you have a paralyzing effect on all of your skeletal muscles, tetanus, botulism, these all paralyze the muscles, which means that the thoracic wall is incapable of pulling out on the lungs. So we've got the resistance in the bronchial tree that's going to affect it, which is going to be most affected by bronchodilations. We've got the surface tension in the alveoli that needs to be combated by our surfactant. And then we need to make sure that both the lungs and the chest wall have compliance. In other words, they are able to do what we're asking them to do. All right, so then if we think about the volumes coming in, okay, so we're going to think about breathing here for a minute. This basically shows all of the different volumes that we could measure. I want you to think about the biggest possible breath that you could take in and breathe back out. And that would be represented by this measurement right here, which is vital capacity. So you're going to take as much in as possible and out as much as possible. Total lung capacity is going to take vital capacity and add on this piece down here, which is residual volume. So let's talk about the three measurements that make up vital capacity first. Tidal volume is this yellow stripe. This is the amount of air that you would be breathing in and out through your nose when you are at rest. It's somewhere in the neighborhood of about 500 milliliters. What you could inhale on top of tidal volume, if you were breathing as much as you could, is called your inspiratory reserve volume. It's there to be utilized if you need it, but when you're just doing normal quiet breathing, you're not going to see that. Similarly, the amount of air that you could exhale after tidal volume is a reserve volume. It's expiratory reserve volume. So you take inspiratory reserve volume, tidal volume, and expiratory reserve volume, you add them together and you get vital capacity, which is the biggest breath that you could take in and push back out. Total lung capacity requires that we add in this measurement down here, which is residual volume. So let's look at these two for a second. Inspiratory reserve volume is pretty big. Expiratory reserve volume is only about half of that. So where is this going then? And the answer is into residual volume. This is an amount of air, a volume of air that cannot be expired, it cannot be exhaled, because it stays in your lungs all the time. So if you take the biggest breath in that you can, you're not going to be able to let all that you took in back out because some of it stays behind as residual volume. There are two very important physiological roles of residual volume. Number one, it's what keeps the alveoli inflated. If you were to fully remove all the air from your lungs, even in the presence of surfactant, that collapsing nature of the alveoli would collapse them closed and enough of the water molecules would be cohesive and you wouldn't be able to get them back open. So in order to maintain the alveoli, we have to make sure there's always air in them. And so that's what residual volume does. The second thing it does is it deals with gas exchange while you are exhaling. Blood does not stop coming into your lungs to get oxygen just because you're in the middle of exhaling. There has to constantly be air in your lungs for continuous gas exchange. It takes roughly 90 seconds of breathing to fully refresh your residual volume. We can measure vital capacity and the three volumes that make it. We cannot measure residual volume because we would have to physically collapse your lungs in order to get the air out. So residual volume is calculated based on these. Now, there are a ton of different variables that can produce different volumes here. Some of them are physical, like your age, whether you're male or female, and your height. These things are not really things you can change. When you're very young or very old, your volumes are lower than in the middle of your life. Males have a higher capacity than females, even if they have a similarly sized body. And height, because it plays a bigger role in the size of the thoracic cavity, also plays a role. Then there are physiological factors that kick in. What do you do with your lungs? Do you never do anything with them? Then you're going to have lower volumes. If you're a cross-country runner or an opera singer or a swimmer or anything that really taxes your lungs and forces them to work, you're going to have higher volumes. So your lungs will respond to physiological training as well. And then there are the negative physiological factors. Do you have asthma? Do you have COPD? Things that would negatively impact it. So this is, the standard anatomy guy version of this. But there are multiple different factors that come into play that would dictate what your individual volumes would look like. Okay, so now we're going to talk about something called dead space. Anatomical dead space is the amount of air that is in the conducting zone. So if we say your tidal volume is about 500, then about 150 mils of that is stuck in the conducting zone. So that means that you're really only being able to use about 350 mils of each breath for gas exchange. So everyone has anatomical dead space. Alveolar dead space is any damaged or diseased alveoli that cannot undergo gas exchange. Add those together, you get what's called total dead space. So if we kind of look at two different measurements, we'll start with minute ventilation. This is literally just ventilation. It's the amount of gas that flows in and out within a minute. So if we take our 500 milliliter tidal volume, multiply it by an average of 12 breaths per minute, you're going to be moving about 6 liters of air per minute. Now, this can dramatically increase during exercise. Alveolar ventilation rate, though, takes into account dead space. This is an indication of how well you are moving air. This one is an indication of gas exchange. So alveolar ventilation rate takes your breaths per minute, and then it takes tidal volume minus total dead space. So this gives us an idea of how much gas exchange is occurring. Both of these are useful measurements. They are just different between the two. Last thing we're going to talk about is the control of respiration. So this is in the brainstem. We have what are called medullary control centers and pontine centers. So the medullary is in the medulla oblongata and consists of two groups, starting with the ventral respiratory group, this little guy right here. This is the primary part of your brainstem that is going to generate the basic rhythm of breathing. So somewhere between 12 to 15 breaths per minute. It's going to alternately deal with inspiratory and expiratory neurons, and it's going to do it in a very basic rhythm that allows you to breathe. Injuries to the ventral respiratory group are going to require you being on a ventilator because you can't create your basic rhythm of breathing. The dorsal respiratory group, you can see, has a lot of receptors coming into it, a lot of input coming into it. Its job is to take input from both central and peripheral receptors and determine if we need to change our respiratory rate to account for anything. The pontine center, this guy right, where are we? Here, okay, is going to override this ventral group to modify inspiratory neurons for specific patterns. When you sleep, when you're talking, when you're exercising, when you're singing, when you're holding your breath to swim underwater. So the pontine kind of modifies this rhythm based on what you're doing. So again, the most critical is this one. This is where you get your basic rhythm. This one is going to allow us to take input from elsewhere and decide if we need to change the rates. And this one gives us the ability to modify based on certain patterns that we are creating. So let's talk about some of the different pieces that play into the changing of respiration. So these are things that are going to go into the Pond Teen Center and pick up this, difference in whether or not we should change. So let's talk about two that would negatively affect respiration first. First of all, the entire system is lined with irritant receptors. If you inhale something that you shouldn't be inhaling, you will cough. You will begin to breathe very shallowly, and you will cough, okay? So if you're in a very smoky room, you don't take big, deep breaths because the irritant receptors are sending information in, and we are changing how we are breathing based on them. Stretch receptors also negatively impact respiration, and this is important. It's going to keep you from overinflating your lungs. If you think of your lungs kind of like a balloon, we don't want to put so much into them that they pop. So the stretch receptors actually inhibit the muscles that allow you to pull out if you're getting to the point of overinflating. Now, positive ones that are going to increase respiration would be the receptors out in the muscles and joints. As you start moving around and your muscles are like, whoa, I need more oxygen, they're going to send that in. It's going to increase respirations. And then we have our chemoreceptors. Central chemoreceptors are actually in the central nervous system, and they are very, very sensitive to carbon dioxide levels, which lead to pH and hydrogen level changes. So we're going to get into this a little bit later, but these two are absolutely related. Peripheral chemoreceptors out in the carotid and aortic bodies pick up any drops in oxygen or increases in CO2, which lead to increases in hydrogen and are going to increase your rate of breathing to try to get us back into homeostasis. The hypothalamus has like emotional pain responses, people who, have hyperventilations when they're painful or scared. And then you've got the higher brain centers, which are going to be your sort of voluntary control. Something to note is that you can't hold your breath till you die, even if you want to. Kids often threaten this. They say they're going to go into the corner and hold their breath till they die because you're mean to them. Yeah, that's not going to happen. They can't. So as long as their brainstem is functioning, it's not going to happen. They're going to pass out and then they're going to breathe. So hopefully that'll help you someday when you have children and they're telling you that. Yeah, just say, oh, okay, go ahead, try it, see how it works for you. They'll pass out, be fine. Of all of these, pH is the most critical. We are most sensitive to changes in pH, and it has a much stronger stimulus in terms of changing respiration than anything else. So it's not oxygen, it's not CO2, it's pH.